welcome also from our side to today's Tech Talk. We would like to start with introducing ourselves and give you a short picture of us, how we look like. So my name is Magdalena. I'm working in Radeberg in Germany as lab microbiologist. And together with me today is David, who works as a senior scientist in El Paso, Texas in the USA. On our agenda today is at first the topic to explore together with you the fundamental principles of bacterial endotoxin testing and to gain detailed insights into the key methodologies and techniques that are employed in this critical testing process. Additionally, we want to understand the disparities and commonalities between the new ISO 11773 standard, as well as the ANSI and AAMI guidelines. And we want to show to you our comparative analysis between the new ISO standard as well as USP regulations. And last but not least, understand the unique approaches and requirements outlined in each and every standard. Let's give you first a short overview of the related standards and references that we will be talking about during this Tech Talk. So, of course, we will refer to the USP 85 as well as 1085, giving guidelines on the 85, and the USP 161 for medical devices. In addition to that, a large fraction of our Tech Talk will cover the ANSI standard, as well as some of the FDA documents that give guidance for bacterial endotoxin testing. At first, let's talk about some key terms that we will use in this Tech Talk and give you some detailed definitions, what we actually mean with them. So the first, of course, most important definition is what is BET? So when we're talking about BET or BET, we mean bacterial endotoxin testing. Other terms which are often used to describe this test include also the abbreviation LAL testing or Pyrogen testing. What is LAL meaning? With LAL, we refer to limulus ammobiotide lysate. This is used to estimate endotoxin levels in BET, and a more common name is just lysate. A pyrogen is a substance that causes fever and in connection to bacterial endotoxin testing, this is lipopolysaccharides or LPS. Next very important term is MVD or maximum valid dilution. This is a term that influences our BET detection limit and is based on the sample dilution and or also the extraction volume used to extract bacterial endotoxins from a product sample. Of course, we want to also give you a short information about LPS. So LPS are lipopolysaccharides. This is a very important component of the gram-negative bacteria cell wall, and we will illustrate this to you later in the Tech Talk more in detail. For bacterial endotoxin testing, we use a special type of water. This is called VBT, water for bacterial endotoxins tests, and sometimes it is more generic, also referred to as LAL reagent water. And yeah, we use this for BET extraction. Method suitability is also a key term. Sometimes it is referred to as well as a so-called inhibition or enhancement test. This is a very important step used within the validation, for instance, but also in routine testing to determine if a sample contains interfering factors that could diminish the accuracy of a BAT. In addition, we have lambda, which is, well, a Greek letter, but also an important key term describing the sensitivity of the lysate in endotoxin units per milliliter, or it can be also referred to as the lowest endotoxin concentration in the standard curve, which is important for the chromogenic as well as the stoichiometric bed testing methods. And last but not least, endotoxin itself. So endotoxin, when we talk about endotoxin, we refer mainly to bacterial endotoxins, which we again already mentioned is a component from lipopolysaccharides in gram-negative bacteria. What unit can be used to quantify endotoxins? Very important unit is given here, which is EU per milliliter, so an endotoxin unit per milliliter, which is one international unit that is used to measure BET. We want to explore together with you more in detail on the role of pyrogens and bacterial endotoxins. So what is a pyrogen again? Pyrogens are any substances that can induce fever, which is very critical in the human body because this could even lead up to death and severe infections. And the predominant pyrogenic contaminants that we find in the manufacturing process of healthcare products are bacterial endotoxins. We have this illustrated nicely here for you, so you can see a little, well, example of a gram-negative bacteria cell. And inside this outer cell wall, we can find the endotoxins, the lipopolysaccharides that are so crucial. Of course, this is a simplified illustration. And in reality, these cells have very complex multi-layered membrane systems, and the lipopolysaccharide itself attached to the outer membrane is consisting of a O antigen, a core structure, and the lipid A. And the toxic component, which is responsible for the most toxicity in this whole molecule, is the lipid A component. So one could ask ourselves now, why do we find gram-negative bacteria in or on healthcare products? Where do they come from? Of course, they could come from a manufacturing process that include microorganism itself. On the other hand, it could be also a manufacturing process that include water sources. And this is actually the most significant and also prevalent source of bacterial endotoxin contamination. In addition, gram-negative bacteria can also be present, for example, inside pipes, in biofilms, or in product components with animal tissue origin. We also have to ask ourselves, how can we now detect bacterial endotoxins reliably and in a sense with a test assay that is sensitive and also very specific and accurate? And for this purpose, we use the amoebocyte lysate reagent that we extract from the hemolymph of the horseshoe crabs and perform our bacterial endotoxin test. As already mentioned, This is a truly highly sensitive test method, and let's say its principle is thanks to the blood cascade, the blood clotting pathway of this beautiful horseshoe crab. You have to imagine that when the horseshoe crab is infected, for example, with some gram-negative bacteria and any endotoxin would be present, this would activate then hemocytes, which cause a release of coagulation factors and antimicrobial substances. So a whole cascade of different enzymes is activated. And at the end of it, we have the formulation of a coagulant gale or simplified blood clotting. And this whole defense mechanism, which occurs naturally, is essentially the principle that we can use in the laboratory for our BET test. Now that we understood the horseshoe crab's role, let's take a close look on all required steps in the whole BET process and start with your role in it, with your production.
Okay, thank you. We're going to take a look at the principles of BET, and it does start with the selection of products. We have a little illustration of over there of some products that would be relevant, and we'll also go through the definitions that we have here. So one is the product's intended use, and this is the clinical application. We can also refer to any national regulatory requirements that are set forth through your regulators. So the product's intended use, as we have seen, can vary, and there's lots of different products that would be involved in this scenario. The other way that we're going to have selection of products is if your product has a label claim. And the label claim here would be a non-pyrogenic claim. The non-pyrogenic claim could refer to a certain portion of the product, for example, a fluid pathway. When we're looking at product selection, there's two scenarios that we can look at testing. We can either look at individual testing or testing the products as a pooled extract. And besides the healthcare products, there's also in-process sampling that can be done. And one of the common sources for in-process sampling are water samples. And these water samples are taken from the production and they're tested alongside your product samples. Continuing on with the principles of BET, with product requirements, we look at the required label claim, or we look at how the product is being used. And there's a couple approaches that we can look at when we're testing products. So first of all, there's batch testing. In batch testing, these are batches that are defined as a lot, and lots can be defined as the customer sees fit. However, you're typically looking at a uniform character or quality that's within a defined cycle of production. When you're developing your batch testing scheme, you also want to document your rationale or do like some sort of risk assessment so that you're understanding how that batch testing is working. The other scenario is an alternative to batch testing. Alternatives to batch testing, again, you would use a risk assessment to define criteria for a sampling plan. And also this can be used after demonstrating that the manufacturing and the materials that are used for the product are controlled. And we would like to note here that your samples can come from either prior to sterilization or after sterilization, so pre or post sterilization. And just a note that when you're looking at pre-sterilization samples, it cannot always be appropriate. You might need to justify pre-sterilization samples. And one scenario that could be not appropriate for pre-sterilization samples are products that support microbial growth. Okay, so moving on here, now that we have selection of product, we can take a little look at a scheme that would be used for BET testing. So first you have product handling. And in this scenario, we have the syringe, and the testing portion that's outlined there is going to be removed, and the testing will commence. So the next step here is we would put the test portion into the extraction solution, the WBET, the water for bacterial extraction. And then we're going to continue with the extraction process for the stipulated time. So then once the extraction is completed, we'll do the analysis portion. And during the analysis, a dilution of the product extraction solution is typically done not to exceed our maximum valid dilution. And then the final step would be the actual assay. And there's 3 assays that we will follow. So one would be the gel clot assay. The second would be a kinetic turbidimetric assay. And the third would be a kinetic chromogenic assay. We'll go into those a little more detail later on. So we're going to take a little bit closer look on product handling and the endotoxin extraction portion itself. So first of all, anything that we're using to handle the product needs to be depyrogenated. This would include tools, glassware, anything to disassemble or prepare the product for the extraction. The other key component here is the product-specific endotoxin limit. We will use this based with the maximum valid dilution to perform this calculation. This influences the BET limit of detection, and we would adjust the lysate sensitivity accordingly. The other portion is how much of the water that we're using for the extraction process and how many steps might be needed to complete the extraction. And the table there gives us an idea of what kind of limits that we're working with. For devices that contact the circulatory system, less than 20 EUs per device is typically used. For cerebral spinal fluids or devices contacting that portion of the body, a much lower limit of less than 2.15 EUs per device. And when you're looking at interocular devices or kits, your requirements are even less, at less than 0.2 EUs. So when we're looking at products, there's 2 different forms. We would have either solid product, which would require extraction with the BET solution or LRW. And liquid products, we would not need to perform any additional extractions. We could test as is and just put through the system. Additional considerations for extractions would be agitation or no agitation. Agitation is always recommended if possible. There's also 2 different routes that we would take, either pre-warmed water at 37 degrees C body temp, and you would be looking at a 1-hour extraction under ambient conditions. Or you would be looking at doing an extraction between 37 to 40 degrees C with your water, and that would be for 15 minutes. Any other extraction times would be justifiable through a risk assessment or other documentation. All right, well, moving on, we will also look a little bit closer at our methods that we use for BET. So there are 3 methods that are generally accepted. The first one that we want to talk about is the limits test, and this one is a gel clot test. These are considered usually qualitative, and it's based on a formation of a clot. So with a clot, you have a positive test, no clot would be a negative test. The second one that we would consider is an endpoint method. An example of this would be a chromogenic photometric method. These are quantitative methods, and it's based on color change. So once the color change has occurred, the optical density is read, and the value is determined through linear regression on a curve. The other third method is a kinetic method, and here we have both chromogenic and a turbidimetric. These are both considered quantitative methods, and these are based on a time to a predetermined optical density. All right, so if we look at a little bit closer, how do we determine which BET method would be appropriate for the product that we have selected? So the choice of technique should be based on the following criteria. One would be nature of the product sample. Two would be the laboratory's capability. With this is also the laboratory's experience, so particular experience with methods. The next criteria would be sample throughput requirements. And lastly would be data handling requirements. So with those requirements, we would have the criteria that we could use to select which method would be appropriate for our test. All right, now that we've have a pretty clear understanding of the details of the bacterial endotoxin testing, we're going to dive in a little bit closer into the ISO standard requirements and other standard requirements and do a little comparison and contrasting with those standards.
Thanks, David. I would like to start to present to you a comparison between the ISO standard requirements and our USP requirements for the different USPs that gives us guidance. And the first point I would like to make is about the limits that the ISO standard is clearly giving. So according to the ISO standard, the healthcare products that contain pharmaceuticals should be tested and evaluated according, of course, to pharmaceutical drug sampling requirements. So this is very logical and makes perfectly sense to say, okay, guidance is, for example, giving in the USP 1085 to with respect to drugs, to biological compounds, as well as combination products. So let's say a combination of a drug and a device. And yeah, another aspect that the ISO standard is considering is with respect to a suitable sampling plan. And this is nicely illustrated in a table in the annex of the ISO standard. And there you can find how many product samples you shall test based on your batch size. And the guidance from the ISO standard here is very, very similar compared to USP 161, where it is stated that not less than 3 devices and not more than 10 devices shall be tested. However, the ISO gives the option to say, okay, for very small batch sizes, testing only 2 products is fine. And another guidance in the USP 1085 would be that the minimum amount of 3 devices or units that are tested for BAT should represent historically the beginning, middle, and end of each batch. A third point I would like to make here is within the section 72 to one of the ISO standard where it is specified that solid healthcare products are either tested individually or pooled. The ISO specifies that for pooling, a maximum of 10 healthcare products is all right and not more. However, there's no background information in the ISO standard on pooling itself. And this you can find perfectly detailed in the USP 1085. Here are also some considerations given and some risks are detailed with respect to the pooling approach. Moving on, there's also a section in the ISO standard about equipment and materials, which is rather general. So, for example, it is stated that the equipment used for depyrogenation of glassware or other equipment in BET testing shall be qualified and maintained, and methods shall be established, validated, and documented. Here, USP 85 would give you more detailed, very specific guidance. For example, it is stated in there that a commonly used minimum time and temperature for depyrogenation is 30 minutes at 250 degrees. Additionally, you can find in section 7.2 of the ISO standard certain aspects regarding the requirements for the negative control, the standard control series, as well as sample solution and product positive controls. And this is identical for the gel clot method, no matter if you take a look into the ISO standard or if you take a look into the USP 85. However, when we take a look at the kinetic or other endpoint methods, the number of replicates that is recommended in the ISO standard for each concentration of the standard control series is only 2, whereas in the USP 85, it is mentioned that you should have at least 3 replicates for each standard endotoxin concentration in your standard curve. Additionally, the ISO standard opens up the option to achieve this standard control series requirements with an archived standard curve. However, there is no detailed guidance given, for example, in the annex of the ISO standards regarding this new possibility. With respect to sample interference, all standards, so the ISO standard as well as also the USP standard, give very, very good guidance what you could do when you observe a sample interference, which means you would likely observe in the BET an inhibition or maybe an enhancement. And the top considerations from the standards are to dilute your product sample extract, but not to extend your maximum valid dilution when doing so, as well as the options to filtrate, neutralize, dialysis, or heat treatment your product sample extract. It is even possible to combine those options that are mentioned here to overcome sample interference. Another aspect with respect to standard requirements is of course to answer the question, when is a bacterial endotoxins test actually valid? And according to the ISO standard, the acceptance criteria for the gel clot method and also for the other methods, so for the kinetic and endpoint methods, are detailed, and those are the same acceptance criteria as we find for these tests in the USP. The ISO standard gives also small, let's say, additional criteria and just points out that one should consider an acceptable maximum for the coefficient of variation within this test assay. With respect to exceeding endotoxin limits in the BAT, all standards are giving very detailed guidance on failure investigation. There's a whole Annex C in the ISO standard, which is very similar, almost identical to the Annex C of the American standard, and gives very good guidance and points out also a lot of aspects that you could also find in the USP 1085 in the section out of specification results and retesting considerations. And with this last detail, I'd like to, I'd like David to take over for more ANSI standard requirements and aspects.
Okay, thank you. Let's take a closer look at the ISO document, and we're gonna dive into this now. So when we look right away into the introduction of the ISO document, we get this statement here that says that it's based on the ANSI AAMI standard 72. However, they do point out that several sections have been restructured, extended, or changed from the ST72. So we'll take a little bit closer look at maybe some of these that could be impactful. The next thing I would like to point out to you guys is in section 122, and it just specifies that there are no specific endotoxin limit specifications, and the document doesn't specifically address those. Although we'll take a closer look at that in a little more detail because I think that's an important aspect later on. But moving right along, we can see the next section that we have some changes here is within the terms and definitions. The ISO has added some terms related to BET testing that we're familiar with in the process, and there are some changes in wording that have been added for some additional clarification. Next, we kind of look at the whole document as one, and we can see that there are very similar intent and requirements throughout the document. And these are found throughout the extraction process for products, the methodologies that we use for testing, how suitability testing is performed to make sure that your product can be used with the BET methodology for an appropriate limit. We see the selection of product and the similarities there. And even within the various annexes, there's either direct alignment or there's very close verbiage that's being used. We do see that restructuring is evident in many of the sections to align to ISO documentation. However, a matching section can be found within the standard 72 with similar content and a similar intent as what we're finding in the ISO document. One thing that I would like to bring attention to is within section 9. It's on the maintenance of the BET method. It goes into suitability for changes on the process or the method. So these changes would be either for the product changes or the method changes. And they just have a statement here that says a one batch suitability study is sufficient for reassessment. Another section that we have that's important to note is the maximum valid dilution. This is within the ISO section 6233. It's in a note for note 2. And there's this note that says that when a product cannot be tested at a dilution less than or equal to the MVD, then there's a risk document that needs to be created in order to justify that. and then also a recalculation of the MVD and the endotoxin limit would be something that you would want to take a look at in a little bit more detail to make sure that the justification is there for that exceeding of the MVD. Okay, moving on. We have a little bit more detail that we go into on the BET limits. We did note that the document doesn't specifically address setting of limits for endotoxin. However, in these various sections that we can see here, this is section 511, uh, limits shall be determined and consistent with the intended clinical application. This is resonates true for both standards, just reminding the user of the document that you always want to look back at the intended clinical use. Also in section 5, you see that there's this caveat where a higher endotoxin limit could be justified. And this would be depending on the risk benefit of the device. And in section 6.2, you have, this is section A6.2, the appendix, goes into more detail on consideration of the applicable endotoxin limits. And it refers you back to the USP 161 to take a closer look at limits that are applicable within the US. Another portion here is in the annexes. We see an addition to the annexes in the ISO document. This is Annex F. It covers the responsibilities, and it lists in a table format the role of the manufacturer and the laboratory as either being responsible or informative. And this is just, it goes clause by clause and informs the reader who would be responsible or who would be providing information in those various portions of the test. Okay, well, we've gone through the comparative of the ISO standard and the USP to standard 72, and we would just like to bring up a couple conclusions that we come to. So first of all, the ISO 11737-3, it brings the BET testing worldwide through the Federation of National Standard Bodies, and it adds to the existing United States-based ANSI AAMI Standard 72 and the United States Pharmacopeia. Additionally, the ISO resonates with harmonization and collaboration and agreement among the BET analysis, and this helps the manufacturers to demonstrate safety to patients on a worldwide scale. And with that, we conclude our presentation, and we will be opening up for a Q&A session. But for more information, please feel free to reach out to either one of us, and we thank you for your attention.